So we just talked about chemical synapses in the previous video, and now we're going to look at what types of potentials are generated in those chemical synapses. So with that postsynaptic potential, you can have an excitatory postsynaptic potential that moves you towards threshold by opening ion channels that allow positive ions to flow in. You can also have inhibitory postsynaptic potentials that allow positive ions to flow out or negative ions to flow in. And so that causes a hyperpolarization. So excitatory causes a depolarization, inhibitory causes an hyperpolarization. And then they sum together to give you an overall change in your membrane potential.
Recall that all action potentials that reach the axon terminal involve depolarization of the same size. How is it then that the effect on the postsynaptic cell can vary? First, the frequency with which action potentials arrive at the axon terminal determines how much neurotransmitter is released. Also, in the nervous system, multiple presynaptic neurons can stimulate one postsynaptic neuron, and their effect will be cumulative. Finally, depending on which neurotransmitter is released and the types of receptors that are present on the postsynaptic neuron, the effect can be inhibitory or excitatory. We will now focus on excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, or IPSPs. Postsynaptic potentials are local potentials in the postsynaptic neuron. If they are excitatory or depolarizing, they will bring the membrane potential of the postsynaptic neuron closer to threshold. This makes the neuron more likely to fire an action potential. EPSPs typically result from the entry of positively charged ions, such as sodium or calcium ions, via ligand-gated cation channels. If enough excitatory potentials occur in the postsynaptic neuron so that its axon hillock reaches threshold, an action potential will be generated. In contrast, inhibitory postsynaptic potentials, or IPSPs, cause a small hyperpolarization, so the membrane potential becomes more negative, moving it further away from threshold. IPSPs generally result from one of two events. One, the loss of positively charged potassium ions due to the opening of ligand-gated potassium ion channels, or two, the gain of negatively charged chloride ions due to the opening of ligand-gated chloride ion channels. IPSPs make the neuron less likely to reach threshold and therefore less likely to fire an action potential. Recall from our discussion of local potentials that a single neuron can receive many incoming stimuli in the form of depolarizing and hyperpolarizing local potentials. If these local potentials are caused by synaptic stimulation, they are called postsynaptic potentials. The neuron then integrates all incoming stimuli, and if threshold is reached, an action potential will be fired to propagate the signal farther down that neural pathway. If threshold is not reached, then the signal will stop at that neuron.
So let's review. So postsynaptic potentials. They're going to be C. Depends, because we just saw they can be excitatory or they can be inhibitory. So it just depends on which channels are open as to whether they will be excitatory or inhibitory. Excitatory postsynaptic potentials are move the postsynaptic membrane towards threshold, so move it towards depolarization. Inhibitory are inhibitory. So they move towards hyperpolarization. So your synaptic transmission may be terminated by ending the effects of the neurotransmitter in one of three methods. So they can diffuse away from the synaptic cleft and be reabsorbed. As we saw with the neuromuscular junction, it can be broken down in the synaptic cleft by enzymes, and the byproducts of that be reabsorbed. And some neurotransmitters are reabsorbed into the presynaptic neuron by a process called reuptake. So these are three ways in which you can get rid of the neurotransmitter, stop the stimulation, and return the membrane back to resting membrane potential. And so here are those three methods: diffusion and absorption, degradation by an enzyme, or reuptake via pumps.
Synaptic transmission is a transient event, like speaking a single word. As you've seen, it begins with an action potential arriving at the presynaptic terminal. Stimulation of the postsynaptic neuron will last as long as neurotransmitters are available to bind to postsynaptic receptors. When the neurotransmitter involved is no longer available, stimulation is terminated and the postsynaptic neuron returns to its resting state, allowing it to respond to future stimulation. We will now examine the three mechanisms that allow for termination of synaptic transmission. The first mechanism allows some neurotransmitters to simply diffuse away from the synaptic cleft. They might eventually diffuse into surrounding neuroglial cells. Another mechanism for synaptic transmission termination is degradation of the neurotransmitter by an enzyme in the synaptic cleft. An example is the enzyme acetylcholinesterase, which breaks down acetylcholine. The components of the degraded neurotransmitter are often recycled back to the presynaptic neuron and then reused to synthesize more neurotransmitters. And finally, other neurotransmitters, such as dopamine and serotonin, are removed from the cleft by reuptake into the presynaptic terminal, a process facilitated by specific membrane transport proteins. Once inside the axon terminal, the neurotransmitters can be degraded by an enzyme or repackaged into vesicles and reused. Regardless of how the neurotransmitter is removed, once it is no longer in the synaptic cleft, synaptic transmission is terminated.
So termination of synaptic transmission is necessary because your postsynaptic response cannot be reinitiated until the first response is terminated. Which of the following is not a method for termination of synaptic transmission? Receptor fatigue, that was not mentioned as one of the three mechanisms. Which of the following is the correct sequence of major events during chemical synaptic transmission? Remember, look at them and see which one happens first. So the first thing that happens is an action potential, then synaptic transmission, postsynaptic potentials, and then synaptic transmission termination. And so here's our big picture of putting it all together. From the action potential to the synaptic transmission to the postsynaptic potential to termination of the synaptic transmission. And how is all this integrated? So with the inhibitory or excitatory signals from multiple neurons, how are they integrated together to generate one action potential? And so we have different ways in which integration takes place. and that is called summation. So the added together effects of our excitatory and inhibitory, whether they reach threshold, will determine whether we generate an action potential. And so we can have different types of summation. We can have spatial summation, and we can have temporal summation. So all of these guys are giving excitatory, and they bring it to threshold, which generates an action potential at the axon hillock. And so with spatial, simultaneous release of neurotransmitters from many presynaptic neurons, like we just saw in the previous figure, or you can have temporal, which would be repeated release from a single presynaptic neuron that would add up to the threshold. And so here is temporal, single neuron, axon terminal, repeatedly releasing neurotransmitter, repeatedly depolarizing, getting closer to threshold with each stimulation until you reach threshold and generate an action potential. And then spatial summation is where you have multiple neurons releasing neurotransmitter at the same time, bringing the depolarization to threshold, generating an action potential at that axon hillock. And with neurotransmitters, some neurotransmitters can be both inhibitory and excitatory depending on which receptor they bind to. And so we've got lots that act in both mechanisms. There's a nice table in your lab manual that summarizes these. Table 11.3 tells you if they're excitatory, inhibitory, and the ones that are both. Have 5 major classes. We have the acetylcholine with acetylcholine, your biogenic amines, your catecholamines like norepinephrine, epinephrine, serotonin, histamine. Those are all biogenic amines. We have amino acids such as glutamate, GABA, glycine, neuropeptides such as substance P, opioids, and neuropeptide Y. And so this tells you whether it's excitatory, inhibitory, tells you the locations and the type of receptors, whether they are ionotropic or metabotropic. Ionotropic acts directly and metabotropic acts indirectly. All right, so that is chapter 11. Let me know if you have any questions.